Hello, good morning, everyone. I hope you had a good weekend, and we are going to get started. So today we will take up the lab PowerPoint, and after that we will do the test review. And if you have any additional questions, today is the day to ask. So this one is available online, and let me just quickly show you where to get it for those of you who haven't downloaded yet. I only saw 10 people accessing the file. So if you just go to the homepage, course homepage, if you scroll down to the second announcement, there's a hyperlink to get to it. Okay, complete online lab 1. So you just click on that and it will give you the PowerPoint file. And while you are at it, you can also download the test 1 review. Okay, so for those of you who haven't got it, or if you're watching this on YouTube, that's where you get the files for the things I'm about to go through with you. So let's get started. This is a heart model, and as the title tells you, it is an anterior view. That means you're looking at it from the front. And we are going to label the numbered part, okay? So over here, A is basically the aorta. Okay. So aorta, the function is to carry oxygen-rich blood to the body. That is the aorta. Now, just to clarify something, in case people are confused, the lab quiz, which will be available today at 10 a.m., and it will continue to be available until tomorrow, 10 p.m. That lab quiz is 2.5%, and it covers everything we learned so far, including the cardiovascular system. The test, which is going to happen on the Wednesday, that does not include the cardiovascular system. Okay, so I'm doing this heart stuff, the cardiovascular stuff, for the purpose of the lab quiz that you will be doing. All right. B, what is B over here? B is connected to the right ventricle, and it is the pulmonary artery. Okay, so this carry O2 poor blood to the lungs for reoxygenation. C, okay. When we do this at school, many students ask me what C is. Anybody figured it out here? What do you think C is?
Coronary vessels.
Yeah, someone says septum, someone said coronary artery. I believe the septum would be inside. You cannot see the septum on the surface. Like if I open up the heart, I think the next one opens it up, right? Like, yeah, like that would be the septum, right? Separating the right and the left side of the heart. This is, perhaps the label is not very well placed, but it's talking about these blood vessels, right? So that is the coronary artery. Okay, and veins, right? So that's for circulation, for heart muscles. Okay, so the heart cannot use the oxygen cannot use the blood from inside its chambers, is going by very fast, very high pressure. So it relies on the coronary network to basically do gas exchange and stuff like that. D here is the superior vena cava. Superior vena cava collects O2 poor blood from systemic circulation. Okay, specifically from the top, like 1/3 of the body, right? So that is that. Stop me if I'm going too fast or if you need clarifications. Okay, this is a heart anterior view, and it is a coronal section or frontal section. You cut it like this, right? We are supposed to label H, J, and M. So H is the right atrium. J is the septum. M is the tricuspid valve. So the function of M, the tricuspid valve, is to prevent backflow, right? Prevent backflow to right atrium. And septum, as I mentioned earlier, it separates the right side of the heart and the left side of the heart, so there is no mixing of blood, right? Remember, the right side is oxygen poor, the left side is oxygen rich. We do not want to mix them up. What are K and L collectively known as? So K is the pulmonary semilunar valve, and L is the aortic pulmonary, aortic semilunar valve. So collectively, they are known as the semilunar valve. And name them. I just kind of did, but let's do it again. K is pulmonary valve. You can add semilunar if you want. And then L is the aortic valve. And their function is to prevent backflow to the ventricles. Okay. Prevent backflow to ventricles. Okay. Pulmonary prevents it going from the right side. Aortic prevents it from going to the left side. I is the left atrium. And N is the bicuspid valve. All right. This is the posterior view now. You're looking at it from the back. E is probably should be a little bit higher up. E is the right ventricle. Right ventricle, and here is the inferior vena cava coming in. Okay. What about G? G is another one that gives people a hard time. Anybody want to tell me what G is?
Is it pulmonary vein?
These are pulmonary veins. Yeah, these are pulmonary veins. And what are they feeding into?
So left atrium?
The left atrium is correct. Thank you. Left atrium. Okay, so this is the chamber. F is the pulmonary artery. Okay, so like, it's hard to show the model without me holding it. So, but then this one, right? This one would go up. The pulmonary artery would go up, and then if you turn this to the back, I mean, you can see this going up as well, right? So this one actually branches left and goes behind this one and comes out here, right? So if I rotate this model like this, that's what I'll be looking at. Okay. So pulmonary vein would be coming in from the lungs, and then pulmonary arteries would be going out to the lungs. Right? So of course, these ones would be oxygen poor going to the lungs, and these ones coming back would be oxygen rich. Okay. So that's the blood typing video that you can watch. It's something that you would have done yourself if we were doing this face to face, but you know, this is the next best thing, I guess. So assuming you have watched that, and we talked about this as well, right? So what type of antibody reacted with the sample? So we're looking for little clumps, right? Remember, when the antibody reacts with the RBC, we call that a cross reaction. A cross reaction will result in agglutination, which is clumping. That's what it means. Agglutination means clumping. And then not in the simulated blood, but like in real life in the body, this would lead to hemolysis, right? Red cells popping. So what type of antibody reacted with the sample? We look for compounds, and the only one that has compound is this guy, right? So antibody B, okay, reacted. No reactions with A, no reactions with RH. What is the blood type? Can you guys type the blood type in the chat box for me? So I know you guys know. Anybody know the blood type? Okay, anyone else? Yeah, so Colleen and Stephanie both said B negative, and they're correct. Very good. So what about this one? Let's try this. What is the blood type here? What antibodies reacted? Okay, Stephanie said A plus, A positive, that's correct. There's a reaction here with anti-A, reaction with RH, no reaction with B. So that's why it is going to be A positive. What about the rest of the class? Does anybody need me to explain this further? Because you will have this question guarantee on the quiz, the lab quiz. So everybody knows how to tell. Fantastic. Moving on. A patient needs a RBC transfusion based on the blood typing result. Is the potential donor's blood compatible with the patient? Right, so we got to figure out their blood type, right? So we have a donor here, and you can see there is reaction with the antibody A and antibody B, but no reaction with RH. So this person is going to be AB negative. On the other hand, there's reaction with RH only. So this one is O positive. No reaction with A, no reaction with B. That's how I know it is O. So is it safe? Is it safe to transfuse AB negative blood to O positive? Not safe. Not safe. Okay. Because the patient will have already has. Okay, so the patient has antibody A and B in the plasma, right? Which will cross-react with antigen A and B on the donor's RBC. Okay, so if you do the transfusion, right, this blood is going to have A and B on it, and then the antibody B and the antibody A will tag it, and it will be a cross reaction. We do not want that, so not safe. All right. Next one.
Question.
Yes, please.
Doesn't the type O patient have no antibodies at all?
No, type O means they have nothing on their RBC, right? So O positive means no A and no B. But they have an RH here. But that's irrelevant for this question. What's important is they don't have A and they don't have B. So your immune system will make antibodies for anything that you do not have, right? So if you don't have A, you don't have B, then you will make anti-A antibodies and anti-B antibodies. Does that make sense? Yeah. Okay.
Thanks.
No problem. Any other question? All right. If you have, you know where to find me. This is a simulation that you can try out on your own. Basically, it helps you explore the relationship between pressure and volume when temperature is held at a constant, but we learned that already. Okay, so basically you would, in the simulation that you can do, you can increase the volume, and then you can see how the pressure changes, and then you can fill in the numbers yourself. So I'm not going to do that right now. You can do it, and then this tells you how to change the volume in the simulation, and then you will plot it. Okay, so the most important point of this exercise is after you plot it, the graph is going to look like what? I'm gonna see if anybody did it. Okay, if you have to describe the graph between pressure and volume, how would you describe it? Did anybody come up with a graph? Anyone had a graph? Doesn't have to be in Excel. It could be just like something you do. Really? No, nobody did the simulation. Did it work? If we click on, let's see if it works. Yeah, it works. Look. So like you increase the volume or you decrease the volume, and then you watch the pressure change, right? And then you plot it, and then you come up with a graph, right? Okay, so Mohammed said inversely proportional. Yep, that's the relationship between pressure and volume. But how would you describe the graph? Is it a straight line, or like how is it? Anybody? No. All of you is just like, please tell me, just tell me the answer. It's too early. It's curved.
I did the simulation, but I did not put it in the graph, but I have the numbers. Yeah.
Okay, that's good. Yeah, and John said it's curved. Yeah, I agree with John. Okay, but is it curved like this or curved like this? Both are curved, right? What do you guys think? Anybody remember like high school?
Second one, the second
one. That's right. Okay, so this is the graph. If I give you, now listen carefully, I'm trying to give you hints here. Okay, if I give you a bunch of graphs on the test or on the quiz or whatever, and I say which graph represent Boyle's law, okay, make sure you choose this one. Okay, this is what it would look like if you plot the numbers. All right, so 3 marks for you. Moving on. Respiratory system now. A. Okay, A here, that is the 하요이봉. 하요이봉. B is the thyroid cartilage, protecting your larynx. C is one that gives people trouble because I mentioned it, but then I didn't really explicitly write it down in many cases. That is your thyroid gland. Thyroid gland. I think I might have mentioned it in lecture 2 when we do autoimmune disease. Right? Thyroid gland here. It could get big, right? If you have autoimmune diseases like Graves' disease. Thyroid gland. It makes hormones that controls your metabolism. D is just lung. Very, very non-extraordinary. And Y here is the aorta again. Okay. So this is the same model with the heart removed, and we can see a lot more things. H here is just the trachea. Okay, so you can see the trachea continues to come down and branches into the left and right bronchus. What might F be? This again usually gives people quite a bit of trouble. What is F? Type it in the box, or you can let me know. What is F? Okay, we'll come back to it. Let's do D. D is the left bronchus. E, that would be the secondary bronchus. Okay, we can say left main bronchus. And what about G? Right. Let's take a look here, right? This is aorta, and then you remove it. You can follow this part, and you can see it continues to come down, right? So G, sorry, it's getting really messy. G is abdominal, well, we just call it aorta. It's not really abdominal yet. And once it crosses the diaphragm, then it becomes the abdominal aorta. Okay, back to F. Come on, someone must know what F is. It's in front of the aorta, and then it's behind the trachea. What do you think that is? Esophagus. That's right. That's the esophagus. All right. If you didn't know before, now you do. I think that's everything. Okay. A is the epiglottis. And the function is to cover up the glottis, to cover the airway when you're swallowing, so we don't choke. B is the hyoid, providing attachment sites for various muscles. This one is the thyroid cartilage. D is the thyroid gland. And E is the trachea. What structure is protected by C? Larynx. Okay. So this is the same model. As you can see, we can open it up, right? This would be like a sagittal, mid-sagittal cut. And here, this is pointing at the vocal cords. Let me change the color back to red. Vocal cords. Okay. G is pointing at the thyroid cartilage again. Thyroid. Actually, no, sorry. G, oops, sorry. G is pointing at the cricoid cartilage. Kind of go all the way around. Your thyroid cartilage would be up here, right? And then H is pointing at the esophagus. Okay. So this is the front, and this is the back. Okay. Anterior. This is posterior. Okay, so this here is the thyroid cartilage, and then the cricoid cartilage start here and then goes all the way to the back. Again, it's hard for me to show you. I wish there's a way for me to, create something that I can rotate on the computer screen. But yeah. This hole is the glottis. Leads into the airway, the larynx. Okay, let's identify everything. A blood smear, right? I probably told you about blood smear. You put a drop on a glass slide, right? And then you use another glass slide to spread it across. Then you add some dye to it. Okay, we do what's called like an H&E stain in most cases, and then the cell would soak up the dye. So what are the these little red balls? Well, you probably know those are the RBC. Okay. RBC. All these are RBC. Not surprising. You have a lot of red cells, right? 45% of your blood volume is the hematocrit, which is the RBC. This would be the white blood cell. Right? So you have a white cell here, you have another white cell here, right? And then you have another white cell. Remember, only about 1% of your blood is white cell. So that's why they are kind of scarce. And platelets. Anybody see the platelets here? Where are the platelets? Someone describe to me where the platelets might be. Can we even see it on this blood smear?
Would it be the little blue dots?
That's right, the little dots right here. There is platelet, there is a platelet, platelet. Okay, it's very, very small. They're cell fragments, right? Okay, so all those little purple dots, they are the platelets. Okay. Identify any abnormalities in this blood smear compared to the normal one on the previous slide. Any suggestions? This one is pretty obvious. Okay, this one is pretty obvious. I would think it's pretty obvious. Anybody want to guess what's wrong here?
I think it's sickle cell.
And that is definitely correct. Stephanie, what gives it away?
The moon crescent red blood cells.
That is
The moon
correct.
shape.
Very good. So these are crescent shape red cells, right? That is a classic morphology for sickle cell anemia, where there is a mutation in hemoglobin and it is misfold, right? And John and Mouche also agree it is sickle cell anemia in the chat. Very good. Next one is a little bit tricky. Let's see. What do you think the problem is here? What's the problem? While you're thinking about what the problem is, there will be no microscope slide like these ones on this lab quiz, okay? All the microscope slides will be at the very end on the last lab quiz. Okay, so like, I don't know if that makes sense, but because there are only a few slides per unit, it doesn't make sense to put it on that lab. Like if I give you this and ask you what it is, then you can tell me it's a blood smear right away, right? Like it's too easy. But if I put it at the end along with all the other slides that we learned, then it forces you to think a little bit. Anyone here? Like something looks weird, right? Comparing this to this. Any suggestions? Even just guess, wild guess.
There's some kind of infection because of the white blood cells.
And what's wrong with the white blood cells? I agree with you. What's wrong with them?
They're multiplying, but the shape of it.
Okay, so shape, yeah. And also, I'm not so too concerned about the shapes, but I'm more concerned about the. Anybody? Look at the white cells here. 1 white cell, 2 white cells, 3 white cells, and now here are too many, right? Okay, so too much. Too many, I guess. I don't know, you can count them, I guess. So too many white blood cells. Okay, so what are some problems? Well, Colleen said maybe there is an infection. Could be infection, right? If we can identify the type of white cells, we might know what exactly the problem is, right? There is too much eosinophil, maybe maybe there is some kind of parasite. If there is too much basophil, maybe there is an inflammation. Okay, but it could also be like leukemia, right? Leukemia, right? Again, it depends on what kind of white cells, and we barely scratch the surface of this thing when we learn about the white cells. We only learn about neutrophils, macrophage and all that stuff. But actually, remember, all these cells, where do all these white cells and red cells come from? Does anybody remember? Where do we mix blood cells in the body? You should know that for the test. Bone marrow, that's right. Okay, so there are these hematopoietic stem cells in the bone marrow that if you go back to the lecture slide, you can see there are many intermediate steps in between before it forms the final neutrophils, right? Even the red cells, right? There are many intermediate steps, right? So people who study blood, right, like hematology, they would know what each one of them looks like, and depending which one is being showing up in abundance in the slide, then that tells you exactly what kind of abnormalities is happening. Like that's my wife's job. Like her job is to look at blood samples and then figure out which cell type is too much, right? And then she could tell you potentially what the problem is. And for us, we just know there's a lot of white cells, and keep it simple, could be infection, could be leukemia. But what's really obvious is there are way too many white cells. Okay, you should only have a few per slide. Okay, moving on. This is a transverse cut, transverse like this way. Okay, and then you're looking at it from the top, and you can see there are two lumen. Lumen is the inside of a hollow structure. So over here, this is the trachea, right? This is the esophagus. Okay, so this is the front of the body. This is the back. And C is pointing at the cartilage, right? The C-shaped cartilage that's protecting your trachea, right? If you move your finger up and down your neck here, you can feel the ridges of the cartilage. Okay, and you can see the trachea is much bigger than the esophagus, and no muscle around the trachea, but these are actually muscles around the esophagus. Okay, we'll learn this in the digestive tract, but there are 4 layers of tissues surrounding the esophagus. Okay, this is if you cut the trachea longitudinally, and then you stain it, then this is what it looks like. This would be the lumen, and that's where the air passes through. And then we talked about in class that the tissue is the ciliated pseudo stratified columna. So all these purple circles, they are the nuclei of the cells, and it looks like there are multiple layers, but it's not. It's just a single layer. If you are to go on higher magnification, and then you track, like you maybe here, you trace the cell, it will go all the way down, and then there will only be one nucleus. Okay, so these are all individual nucleus of different cell. It's just the way that they're arranged. It looks like they are multiple layers, but they're not. Okay. So B is pointing at the cilia, and A is pointing at the goblet cells. Goblet cells. Of course, the goblet cells are important for making the mucus, which traps like the bacteria or dust particle, and then the cilia will get rid of them. Okay. Blood vessels. So here, this is the artery. This is the veins. You can tell because arteries are thicker. They have thicker walls because they need to withstand the high pressure. And then the lumen is typically smaller. Okay, and then the veins are bigger. Now, like you might be wondering, shouldn't the veins be like circle and not like, you know, kind of flattened? That's just because of how we prepare these slides, okay? So like you will actually have to take the blood vessels and you have to put it in wax, like, you know, candle wax. You melt the wax and then you put this thing in, and then you let the wax become solid again. And then you put it on this thing called a microtome, which is a really, really sharp blade that cuts it in very, very thin slices. So imagine like you have your blood vessel like this, and then when the blade comes down, it might like squish it a little bit. That's why it looks like this. Okay, but rest assured, in the body, it is like fully propped up, full circle. Okay, let's try one more. What is this one? Artery or veins? Artery or vein? What do people think? Artery, vein, yeah. So this is like thicker, right? And then you have some smaller vessels, could be arterioles, could be venules, hard to tell without higher magnification. Artery here, vein here. Okay. So you can see there is still some blood that hasn't got washed out during the preparation, but that's what they are. And that is it for lab 1. Okay. So I encourage you to go do the lab quiz perhaps right away after class, because it's still fresh in your memory. And you know, if you don't do well, that's okay. Study a little bit and then try again, because you have 2 attempts and only the best result will count. Any question about the lab before we move on to our test review?
Yeah, just I want to ask about the lab quiz. So this will be, I mean, little bit similar to this one, online lab, which one we did.
Yeah, so there will be pictures from the thing we just did, but there will also be multiple choice questions. Okay, those multiple choice questions are simpler than what you might see on the test. They're there to serve as a review for the test as well. So you know, there are both diagrams as well as regular multiple choice questions. Okay. All right, this is the test review. Let me just do a quick poll and see what am I dealing with here. Did you have a chance to do this test review? If you did it, if you did most of it, press A. If you didn't do it, press B. Did you do it? Okay, well, thank you for the honesty. It is an overwhelmingly 100% no, which is fine. I know you guys are probably busy. Do all of you have a lot of assessment this week? I would imagine so, right? Because next week is engagement week. So I think you guys are packed with assessment this week, right? Yeah, like the whole cabal, right? Like math, chemistry, and whatnot. So that's okay, right? I'm glad you came to class, even though you're very busy. So I'll try to make this worth your while. All right, so test 1 review here. But
where would we find this review?
Yeah, okay. So
You
if you don't
post
have it, just go to eCentennial right now on the homepage. And then the announcement. Okay, there it is. The test 1 review. Test 1 review. Okay. So for those of you who don't have it, you know, maybe I give you guys a minute or 2 just to get that. Okay. Meanwhile, I'm gonna go run to the bathroom quickly. And I'll come back in about a minute or so. Okay, you guys can go get the file if you don't have it. Okay, here we go again. So hopefully you have that right now, and we are going to review. So this is fractionated blood. Fractionated is just fancy way of saying separating the blood into the different layers. Okay, so we have the top layer, that is of course the plasma. Okay, the plasma. So plasma, which is You have a question, Colleen? I see
No.
your mic. No? Okay. I just saw you unmute. Yeah. So plasma, which is mostly water, right? Approximately 55% of blood volume. Okay. So there are dissolved minerals, nutrients, and electrolyte. Okay, so like you might have some glucose, amino acid, right? Sodium, calcium, so on and so forth. And then what else do we have? Well, there are going to be plasma proteins. Plasma proteins. Okay. So example of plasma proteins would be things like clotting factors. We know what they are now, right? Clotting factors. There are the fibrin, there are the prothrombin. There are the fibrinogen, and you should definitely know that pathway, right? Of how one activates the other. And there will be a question like that on the test. Okay. There are also immune proteins. Immune proteins. Okay, so we know there are antibodies now, right? Antibodies. There are complement proteins. Complement proteins. Okay. And you know, there are other signaling proteins that are important for the immune system. Okay? So these are the main stuff that you will find in your plasma. Okay. Here, this is specifically looking at this like cloudy interface, right? That's the Buffy code. Okay, that's where you will find the white blood cells and platelets. Okay, so as you know, there are many different white cells. I'm not gonna list it again because I think I have another question coming up later. But white cells would be approximately 1% of your, well, I should say these things makes up about 1% of blood. And finally, down here, this is your RBC. So the RBC, when you separate it like this is also called a hematocrit. Okay, which makes up approximately 45%. Okay. So while we're here, let's review some characteristics of RBC. Right, they contains hemoglobin, right, which is an iron containing, I should say a protein with iron in it. And then hemoglobin is important for transporting gas, oxygen. Right? Biconcave shape. The RBC is broken down at spleen and liver, right? And approximately 120 days lifespan. Does anybody remember a hormone that will increase your RBC? What hormone is secreted from the kidney to increase RBC production? Anybody?
Is it histamine?
Good try. Histamine is going to trigger inflammation, so not quite, but good try. EPO, okay, erythropoietin from kidney will increase RBC production. Okay, so remember, if I ask you what increase RBC production, EPO. If you move to higher altitude and stay there for a week, what hormone will be released? EPO. Okay. Moving on. Here is an example of an application question, the ones that people are concerned about. I have two test tubes here. One is showing, one is normal, one is not normal. Which one is normal? Let me launch a poll and see if the class can tell which one is normal. Okay. If you think the left one is normal, please choose A. If you think the right one is normal, please choose B. Which one is normal? Where are the rest of you? Come on. Click that choice. All right. Most of you, yeah, you know, almost 90% of you said the left one is normal. Yeah, that's the normal one. The plasma, you see the plasma? The plasma is supposed to be clear, right? Clear yellow. Okay? So this one is the abnormal one. Okay, this is the one with the problem. This is abnormal. What's the problem? Anybody?
Hemolysis, the cells.
Excellent. Thank you. Thank you. This is hemolysis. Very good. Okay. So that is what happens. Hemolysis means your RBCs rupture. When it ruptures, all the stuff comes out. You know what makes our blood red? Is the heme and the hemoglobin, right? Okay. So when the red cells burst, then you know all those heme got released and then they are lighter now. So it goes up. Right? So it causes the plasma to turn red. This is bad news. Okay. So that's the answer, hemolysis. What might cause hemolysis though? Any explanation? Any suggestions why we have why we might have hemolysis? Go ahead, Colleen.
Hemolysis will be caused by actually just shaking the tube too rough.
Oh, okay. I see what you mean. But I'm talking about like in the body, right? Like let's assume nobody did anything wrong. Technically didn't do anything wrong. Like they didn't shake the tube or whatever. But even like you would have to shake the tube like really hard to break it. Okay. I don't think that's a concern. Like what about the body? Like something is happening in the body that causes hemolysis. Anyone?
Injuries?
Okay, could be. Anyone else? Something that we talked about, guys. Could be injuries, but something that we talked about extensively in lecture 2. Really? Oh, I'm heartbroken. Nobody know. Wrong transfusion, right? For example, right? Wrong transfusion could be, you know, hemolytic disease of the newborn, right? Okay, because of the antibody and the RBC crossing over, resulting in hemolysis. Remember, we just talked about this on the lab, right? Are we clear? Okay. There are other, of course, there are like a whole bunch of other reasons why you might get hemolysis. But that's the one that we are talking about. Okay, I mean that's the one that we've learned. Okay, with the transfusion mismatch. Okay, other reasons for hemolysis is some viruses. They are hemolytic virus, which means they target your red cells and cause them to burst. Okay, things like Ebola cause massive hemolysis throughout the body. And there are certain kind of group A strep, for example, streptococcus could also cause hemolysis. So a bunch of reasons, okay? But for the purpose of the test, if I show you a picture of this and I ask you what is going on, please, hemolysis. Next one, match the parents with their babies. Okay, there will be matching questions. Let's find out. So remember the 4 rules. O cannot have AB kids. AB cannot have O kids. Negative, negative must have negative kids. And then 2 O's also makes an O. So let's take a look at here. I will start with first one. Okay, so first one, we have 2 negative parents, which means this is not their baby. And then we have an O parent, which means AB cannot be their baby. So is it I or is it K? We don't know. I'm going to put it down here first, and then hopefully we can cross out one or the other later on. I will not work with this one. I will want to work with everyone who has O or AB first to make it easier. Okay, this one looks like it's easy to work with. AB, so no O. O parent, that means no AB. Double negative means no positive. So K is their baby. Okay, so K is gone. And if K is gone, then I is the baby for the first couple, right? So these 2 are gone. Am I making sense here? Any questions so far? Wonderful. Next, let's see. Okay, so B negative and O positive. O cannot have AB. So J is theirs. And then this is H. So that is that. Okay. A question that you might have is, how can 2 positive people have a negative baby? And again, you have to think about positive doesn't necessarily means homozygous, right? They could be positive / negative, right? If both of them are heterozygous, as in positive / negative, then there is going to be a 25% chance that their kid will be negative. Okay. All right. Here is another question. I'll give you a moment to try that first. You know what? I'm gonna try to do a breakout room, and let you guys maybe discuss on this and see if it works. Does everybody have this document already? I hope you do. Okay, so this is the document, and assuming you have the question. So I'm gonna do a breakout room. I'll let you guys discuss it. And I will come back and gather results. Okay, please say something in the breakout room. Okay, don't just sit there and not talk and make it awkward. Okay, I'll give you about like five minutes to talk about this. Okay, go ahead. Joan, Shamira, Sutana, and Tesha, did you all receive the invitation to go into the breakout room? You have to accept to go in it, okay? Okay, welcome back. Still waiting for one person to come back. I hope there was some discussion going on. Anybody want to share what they discussed in the breakout rooms? What would the answer be for the first one? Anybody want to share? Please share.
So I figure it would be, since F1 is dominant over F2, it would be F1 and F1, F1 and F2.
Okay, so F1, F1, we call 1, 1, and then
F1, F2.
F1, F2, and then
we'd have F2, F2,
mm-hmm,
F2, F3.
mm-hmm.
And then we'd have just F3, F2, F3, F1.
F1, F3, yeah.
F1, F3,
Uh-huh. And then
F3, F2, and F3.
we have F2, F3 already, yeah. And then 1, 1, 3, 3, right?
And 3, 3, yes.
Okay, good. Now, so these are all the possible genotype. And these are all the possible genotypes. Okay, what about phenotype? How many phenotypes do we have? What are some answers from different groups? Anybody?
Would it be 12?
Okay, 12. Anyone else? Thank you, Colleen. Anyone else? All right, you guys are making me nervous. There will be questions like these on the test. We better understand this. Does everybody understand the genotype, how we come up with the genotypes? Right, because there are 3 alleles, right? F1, F2, F3. So you combine them in various ways, and then you get this. Okay, 6 possible genotypes. Phenotypes. Phenotypes is the expression of these genotypes, right? So how many phenotypes? Okay. Well, F1 is dominant over F2 and F3. That means if you have 1 and 1 together, it's just going to be 1 that's being expressed. If you have 1 and 2 being together, 1 is going to be expressed because 1 is dominant over 2, right? If you have 1 and 3 together, 1 is going to be expressed. Okay, so 1, 1, 1, 2, and 1, 3 will all be the same phenotype. Let's call that fluffy 1. Okay, and then 2, 2 would be fluffy 2. 2, 3 would also be fluffy 2 because F2 is dominant over F3. And then finally, if you just have 3, 3, then it will be fluffy 3. So there are 3 possible phenotypes from the 6 genetic combinations. Okay, 3 phenotypes. Does that make sense? If you have a question, now is the time to ask. Don't be shy. Any question? Okay. So what about next one? What about next one? Okay. If all 3 alleles are codominant, what would the phenotype be? Okay, I'm going to do a poll since none of you are saying anything. Okay, go. This is for part B. If all 3 alleles are codominant, how many different phenotypes are there? And if you're not sure, you can choose F. So I know you're not sure.
Professor, is this review going to be posted? I missed the first half. I had to drop my son to school.
Yeah, I'll post the video. Are you talking about the recordings?
Yeah, this recording here.
Yeah, it will be posted. Yeah, don't worry. It's in the usual place that you find all the recordings. Okay.
Okay.
Okay, let's see. Codominance. What is the meaning of codominance? Codominance means both are being expressed, right? So let's write out our combination again. The genotype doesn't change. The genotype is exactly the same. So 1, 1, 1, 2, 1, 3, right? 2, 2, 2, 3, and then 3, 3. It's just the combinations of these three alleles, right? And you get all these. So 1, 1, right? That would be like fluffy 1. Right, we'll call it fluffy 1. 1, 2, right? Now that they are codominant means both will be expressed. It's like having IA and IB. Your blood type will be AB, right? Because one doesn't cover the other, right? So this one would be 1, 2. Right? This one would be 1, 3 for the same reason. This would be 2. This would be 2, 3, and this would be 3. So 1, 2, 3, 4, 5, 6. There will be 6 different phenotypes. Okay. So depending on the relationship between the alleles, the phenotype might be different. Okay. So read the question carefully on the test. Right. See what's codominant, what's dominant, and then you can choose the answers. Okay, any questions before we move on? All right. Next one, find the genotype of question mark in the pedigree below. All right. So we will start with people who are old or AB. So old is II. This is II. This is IA, IB. And then the rest of the people, this would be IA something, IB something, IB something, IB something. If the kid here is little I, then one must came from each parent. And so this little I must have came from this, the mother, and then the other little I must have came from the father. Okay. And then you see that the grandparents here on the father's side, they are both IB, they both have one IB allele. And so in order for this person to have both A and B child, right, then this is going to be IB. Okay. And on the other hand here, to have the A child, this must be IA. Okay. It's not possible for this person to inherit the IB because then they would not be able to produce the A child, right? Okay, so the A has to come from the mother side, right, which is from here. Okay, and then we cannot tell for sure whether whether the grandparents are heterozygous or homozygous. They could both be IB small I. What we do know is at least one of them must be IB small I. Okay, we cannot tell which one it is, but at least one of them has to be. So those would be the genotype for the question marks. Okay.
How did you come up with the small I with the IB?
This one?
Yeah.
Yeah, because they have a daughter that's type O, right? So
okay.
Yeah, okay. And for those of you who are wondering the same thing, one I is inherited from each parent, right? So that's how I know.
Okay.
Yeah. All right. Multiple choice question. Go ahead. give you a moment to try that. Number 6.
Is there 2 possible answers for question 6?
I don't think so. I don't think so. Choose the best one first, and then afterwards tell me which other one you think it is, okay?
Okay.
Only 5 people have answered, and it has been a minute and a half. I'm gonna give you another half a minute. Hopefully I can log more answers. General rule is don't spend more than 2 minutes on a question on the test. I come back to it later if you don't know yet. Otherwise, you will run out of time. All right, only 7 of you answered, and here are the results. So no reaction with antibody A, no reaction with B, and no reaction with RH, which makes this person O negative. Okay, and so Sarah does not have any ABO or RH antigens on her RBCs. That's why she is O negative. Okay, the test is not defective. It's okay that there are no clumping at all. And then none of the antibodies in Sarah's plasma reacts with the antibodies. We are talking about antigens in Sarah's RBC reacting with the antibodies. This is not about antibodies reacting with antibodies. And as I told you, it's O negative, so not positive. If it's positive, there would have been clumping with anti-RH. Okay. Does that clarify your other choice, Colleen?
Yes. I just thought with the O positive, that's why I know I choose D, but I was saying the O positive, but then I remembered the RH wasn't clumping, so.
Very good. Okay, select all the safe blood transfusion. Okay, let's do this. Hopefully it's not too difficult. All the safe ones, okay? So there are more than one possible answers.
Oh, I think I answered that wrong. I answered.
Oh, that's
one
okay. I wouldn't
is
know
not
it
the
was
safe
you.
one.
Yeah, it's okay. You can just make a change in your notebook, okay? These are anonymous. And I am not joking when I said put this on a sticky tag and stick it to your monitor. It will help you on the test, okay? It's okay, it's not cheating. I allow you to do this, okay? I give you permission to do it, okay? So use this chart, it will help you with these questions. Okay, so kind of all over the place here in terms of the choices. So let's see. O positive to A negative, that is not good, okay? Because only negative can be given to positive, right? Even though it's safe to do it once, but we don't want to do it. It's not a good thing to sensitize people. This one is okay. AB can give to AB, negative can give to positive. A positive to AB positive, that is also okay. That is also okay. Because you see, A can go to AB and positive can give to positive. B negative to AB negative, that is also okay. B can go to AB and the negative can give to negative. And last one is not okay. So the correct answers are B, C, D, which is what most of you have chosen. Very good. All right, next one. Macrophage. Let's classification of macrophage. So macrophage is a type of a granulocyte. Right? They came from monocytes, right? And then they are also phagocytes. And very special phagocytes because they are APC, antigen presenting cells. Main functions, they would do phagocytosis. Okay. Recognize PAMs and eat them. Also very important is that they will activate naive T cells. Okay. They can do antigen processing and antigen presentation. All right. Neutrophils. Neutrophils is a granulocyte. You can see their granules clearly. And they are also phagocytes. Main function is just phagocytosis. Okay. Kind of like the first line of defense inside the body when the barriers are breached. Mast cells and basophils. These are granulocytes. Mast cells are anchored to tissue. Basophils are kind of moving around. So they initiate inflammatory response. Initiate inflammation. Right? Release histamine. Mast cell is local. Basophils, they are more systemic. So if you have like a allergic reaction, that's basophil. Local, like if you have hives, right? Those bumpy things that sometimes come up on kids, like here and there, that's mast cell, localized. Okay, so allergic reactions and inflammation. NK cells, it is part of innate immunity. Okay, these are also all part of innate, okay, part of innate immunity. Innate. Okay. And what do they do? Well, NK cells, natural killer cells, they will recognize and kill infected cells or cancer cells. Helper T cell, this is part of the adaptive adaptive immunity. Okay, and it's part of the cell-mediated immunity. What do? Helper T cells help coordinate immune response, right? They kill infected cells with perforin. And these are the things that will punch holes in the cell membrane and inject toxins to trigger apoptosis. All these are in the PowerPoints, of course. This is just like summary for you. Plasma cells.
Wouldn't the helper T cells also activate the B
Oh,
cells?
yeah, you know what? You are right. I actually made a mistake. Good catch. They don't kill. That I'm thinking about killer T cells. So helper T cells, they don't kill. They activate. Thank you for correcting me. Activate naive B cells. Okay, good catch. The one that does the perforin stuff, that's the killer T cells. Okay, plasma cells, they're part of adaptive immunity. And instead of cell-mediated immunity, they are humoral immunity. Okay. And what they do is they mass produce antibodies. Okay. So of course, those are not the only cells we've learned. There are a lot more. So feel free to, when you study on your own, feel free to add more. Okay. Moving on. Okay, another multiple choice question. Go ahead. Oh, sorry. I think this is a multi-select question. I think I launched the wrong poll. Let me launch again. Sorry about that. Okay, let's give that one more try. All right. How would you contrast specific and non-specific defense? Specific immunity works only after non-specific defense have failed. Yeah, that's true. Okay? You don't need to produce antibodies like all the time for everything that you come into contact with. Most of the time, the non-specific defense will get rid of it, the NK cells, the inflammation, and that stuff. Non-specific defenses and specific immunity requires a prior exposure to be effective. That's not true, okay? The specific, like the specific immunity, actually neither of them require prior exposure, okay? Like it would be more potent for specific immunity if you had prior exposure. Like if I got chickenpox before, then the next time if I get chickenpox again, I'll be able to like kill it right away before any symptom shows up. It's more potent, but it's not a requirement for specific immunity to work. Think about it. If in order for specific immunity to work, you need prior exposure, then it would never work in the first place, right? Because we are not exposed to anything when we are born, right? So this is a contradictory statement. This is not true. B cells and T cells both work in non-specific and specific. That's not true. They are both exclusive for specific immunity. Specific immunity changes throughout a person's lifetime, but non-specific defense does not. That is true, okay? As you get exposed to more pathogens in your lifetime, as you get immunized, your specific immunity will change because now you have more memory T cells for those things, but non-specific defense will remain the same. The barriers, the inflammation, those don't change. Okay, next one, another multiple choice question. This one is a single answer, I believe. Go ahead. All right, let's take a look at this. A RhoGAM injection is an example of artificial active immunity. Well, RhoGAM is not to give you immunity, right? RhoGAM is to give you, it's to be given to Rh negative mothers to prevent sensitization. So RhoGAM is not a type of immunity. Okay, so automatically these two are not correct. And you know, if anything, if you must classify RhoGAM as a type of immunity, arguably it would be artificial passive, because then you're receiving anti-Rh antibody from an artificial source, right? So A and B wouldn't be correct. Nursing an infant to provide the antibodies in breast milk is an example of natural active immunity. That's not true. It should be natural passive. Catching COVID-19 and recovering from it should give you natural active immunity. That's the correct answer. Receiving mRNA vaccine, that is getting artificial, active immunity because you still have to make the antibodies yourself. So the correct answer is D. Name the cell uses perforin to put holes in infected cells. That is the killer T cell. Killer
Oh,
or
sorry, I just have a question.
yep.
So you said nursing an infant to provide antibodies is natural
passive. Yeah.
Oh, okay.
Yeah, because passive is like you did not make, in this case, the baby did not make the antibodies, right? The mother did, right? So that's why it's passive.
Okay, thank you.
Yeah, no problem. Releases histamine to initiate local inflammatory response. That is mast cell. A type of APC that activates naive T cell. Macrophage. Kills cancerous cells, NK cells. Forms blood clot, platelets. Most abundant WBC, neutrophils. Okay. Give examples for each of the following. Formed elements. Formed elements. What are some formed elements? Can we all chip in and say something? I mean, for this to be a review, like you should be coming up with the answers, right? Otherwise, you might as well just read the notes. Form elements. Give me some form elements. Okay, thank you. Yeah, in the chat, it's going on now. Okay, so RBC, WBC, and platelets. Excellent. Okay, next one. Granulocytes. What are examples of granulocytes? Granulocytes. What is it?
Neutrophil.
Yep. Neutrophil. Basophil. Very good. One more. One more. Anybody? Eosinophil. Yes. Okay. Lymphocytes. I'll do this one. There are T lymphocytes, T cells, and B lymphocytes. Coagulation proteins, we did it earlier. Fibrinogen and prothrombin. I mean, there are many, many others, but these are the only two that we've done. Okay, autoimmune diseases. Suggestions? Anybody? Autoimmune diseases.
Lupus?
Sorry, did you say lupus?
Yeah.
Yep. Okay, anything else?
Would sickle cell be autoimmune?
Which one, sorry?
Sickle cell?
No, sickle cell anemia is a genetic disorder that mutation. Yeah, good try though. So, someone else? What are the examples we use in class? Anybody?
Rheumatoid arthritis?
Yep, that's another one. Rheumatoid arthritis. I'm not sure if I spelled that right. Sorry. Okay. That is that. Okay, and then we talk about like multiple sclerosis, we talk about Graves' disease, and a bunch of others. Physical barriers. Anybody? Oh, this one is easy. I guess I'll do it. There is the normal flora. There is the, skin, right? Mucous membranes. Okay, what about immunodeficiency? Anybody? Okay. I found the proper spelling. RHEUMATOID. Okay. Immunodeficiency. Yes. Mohammed said AIDS. Yeah. And then the other one, skid, right? The bubble baby, bubble boy baby. Lymphatic organs. Well, there are a bunch. Spleen, thymus, right? Tonsils, to name a few. Artificial passive immunity. We had a bunch of examples, right? So this would be like a serum. I remember like the rabies serum I got, that's artificial passive. Natural passive, breastfeeding. Breastfeeding. Characteristics of inflammation, redness, swelling, warmth, and pain. Okay. Let's try this one. Launch results. Go ahead. Okay, most of you have answered. Yeah, I mean, if you have been attending the tutorial, you would remember I said the dominant string changes every year. That's why we need to get a different flu shot. Okay, this is another one. This one is a little bit weird, but let's see if you get it. Okay, anyone else? Maybe a couple more seconds. All right, so what's going on here, right? We are trying to teach a T cell how to fight the cancer, right? So you take the T cell out. This is an actual thing, right? That people are doing. You take the T cells out and then you change them in a way that they will recognize the cancer cells. And then you put them back in, and then they will be able to find the cancer cells and kill them. Yeah, this is called immunotherapy. So what must happen? Recognize an antigen that is found only on cancer cells and not other somatic cells. So that is important because you wanted to attack specifically the cancer cells, but not the somatic cells, okay? Recognizes an antigen that constantly mutates in the cancer. That would be ideal, but it's not possible because the T cells, remember each T cell, when I explained this concept to you in class, remember the naive T cells, they all only have like one receptor, right? So that receptor is only good for recognizing a particular antigen, right? So if that antigen changes because of mutation, say on the cancer cells or on like a pathogen, then that specific T cell that was chosen would not be able to recognize it anymore. You would need to have a different T cells. So this would not be possible, right? So for this to work, the receptor must do all of the following except for this one, okay? This one cannot be done, right? It's not possible. One receptor is one key for one lock, right? Trigger clonal expansion, you need to do that, right? You cannot fight it with just one cell, so that is correct. And produce effector cells that will attack the cancer cells. So the correct answer for this one is B. This is the one that will not happen in this particular type of therapy. Okay, I hope that makes sense. If not, please ask some questions. Okay, next one. This is what is this? Answer each of the following question based on the diagram below. What is the expiratory reserve volume of this person? Okay, so expiratory reserve volume is from here to here, right? This is the expiratory reserve volume. And what is that? I mean, it's not the best graph here. On the test, I will probably give you a better one. Okay, so if I have to say this is like half of a half, so more like 1, 2, 5, 0, and then this would be like 2, 2, 5, 0, right? So if you subtract 2, 2, 5, 0 and 1, 2, 5, 0, that gives you 1,000 milliliters, 1 liter, right? Make sense? Okay. You see that this is like half of a half, right? Half is 1,500, and the half of that it's 1,250. What is the label for B? The label for B. Over here, this is B. Goes all the way from here to here. Anybody remember what B is? We learned that just last Wednesday. What is that? From here to here. Good try. Not IVR. IVR is here. That's C. From here to here. That is vital capacity. It's the maximum volume of air that you can voluntarily move in and out of your system. Okay, describe the breathing pattern for a person. Right? So this is just regular breathing in and out. Okay, and then the person exhale as much as they could, and then go back up to normal. And then they inhale as much as they could, and then exhale again. Right? And then once again, inhale as much as they could, and then exhale all the way until they can't do it anymore, and then back to normal breathing. Okay. A is the residual volume. As we discussed last week, it is high in CO2, dead air, not useful for gas exchange. Okay, just a couple more questions, and then you are free to go. Draw a flow chart to illustrate how inhalation and exhalation occurs. Okay, so inhalation. So what happens is rib cage moves up and out, and simultaneously at the same time, diaphragm contracts, flattens, moves down and flattens. And then you would have increased lung volume, right? Because the pleura, right? So the movement of lungs is tethered to movements of rib and diaphragm because of pleura. Okay, so the visceral pleura and the parietal pleura, they're stuck together because of the pleural fluid. So when one moves, it sucks the lungs with it. So it will increase the lung volume, which would then decrease lung pressure. Right? You should know by now, this is Boyle's law. Boyle's law. Okay, and then what happens is when PI is less than PA, right? Air enters lung, right? Due to pressure gradient. Okay. Fluid, right? In this case, air goes from high pressure to low pressure. So if the atmospheric pressure, the PA, is bigger than the intrapulmonary pressure, then it's going to go into our lungs, and then it fills up the lung. That's inhalation. Okay. Exhalation is the opposite. I am confident that you can write it out yourself. Okay. You just change the words, right? Ribcage moves down and in, so on and so forth. Okay. Couple more. Multiple choice. I'll just do this with you. During swallowing, the larynx move up against the epiglottis. The epiglottis move downwards to seal the esophagus. The pharynx move upward against the epiglottis, or the trachea moves upward against the epiglottis. Okay, so it is the larynx that moves up. Right? I told you, right, to put your hand here, swallow, you will feel that it goes up. Okay, so that's the one. Next one, I want you to try. Correct answer is C. Okay, remember, the surfactant is an oily substance that is secreted by the septal cells in the lungs, and their function is to break the surface tension. Surface tension is what's squeezing the alveoli, right? So by breaking the surface tension, it allows the lungs to be more compliant, higher compliance, right? And that helps with the inhalation. Okay, last one should be fairly straightforward. Go ahead and try that out. Okay, so overwhelmingly B, it is the increase in carbon dioxide that triggers the breathing center in the medulla oblongata, right? The brainstem right here, and that initiates breathing. And that's it. That's it for the review. I hope it was helpful. Of course, you can still email me if you have other questions. Good luck with all your assessment this week. I know you guys are super busy, but just a reminder for my course, you have to do the lab quiz by the end of tomorrow, 10 p.m. That's the deadline. And then for Wednesday, you have to do the unit test, okay, which is available from 10 a.m. to 10 p.m. All right. And no class next week. It's engagement week. I will see you the week after on the Wednesday. I'll stick around for a couple minutes in case people have other questions. If not, then again, good luck and thanks for coming. See you all next time.